talk to you guys a little bit about what I have been doing in my postdoctoral work. So I have been a cancer biologist in all my training. My PhD was focused mainly on colon cancer and colon cancer stem cells, and then in my postdoc, I started studying breast cancer and breast cancer stem cells. So although my talk is very breast cancer stem cells, frankly, for the students, these principles can be applied to almost any system, normal and cancer, and to study stem cell biology and to elucidate heterogeneity that we see in tissues. So why am I focused on breast cancer? So breast cancer is one of the most common causes of cancer in women. In fact, 1 in 8 women will be diagnosed with breast cancer in their lifetime. And this year alone, 42,000 women in the United States will die of breast cancer. The death due to breast cancer is primarily because patients develop metastasis. That is when the tumor cells go to a distant organ and start forming tumors there. In the case of breast cancer, it's mainly the bone, the brain, and the lung. They develop resistance to therapies, because of which there is relapse. And all these causes can be attributed to mainly one underlying cause, which is tumor heterogeneity. So what is tumor heterogeneity? If you think of tumor cells like gumballs in a gumball machine, and if all of them are exactly the same color and they taste exactly the same, that is when all the tumor cells are the same. But as my son will tell you, this is not the case. All the gumballs are taste different, and that's why he needs to get exactly the one he wants. And that's what tumor cells are. They are slightly different from each other. And this is not just true for tumor cells, but it's true for almost all cells in our body, and they are slightly different from each other. Like, for example, in the blood, you have the white blood cells and the red blood cells, and you have the stem cells in the bone marrow and progenitors, which give rise to the differentiated cell types. So what I'll briefly do is introduce the system that I study, which is the mammary gland and breast cancer. So if you look at the mammary duct, it is basically 2 layers of epithelial tissue. So you have the inner layer of luminal cells and the basal layer of myoepithelial cells. And the luminal cells are the ones which will produce the milk, which will undergo an expansion during pregnancy to produce the milk. And the basal cells are the ones that will, in fact, help in pushing the milk out to feed the baby. So the mammary gland is also a very unique system because most of the development occurs postnatally. So this is a mammary gland from a 4-week-old mouse, which has not undergone puberty. So you can see that the tree is a very small tree. And as the mouse undergoes puberty between 4 and 12 weeks, this mammary gland undergoes an expansion to develop the mature tree in the fat pad. So just to clarify, the tree is this dark purple structure that you see in the back here compared to the light pink in the background. So the mammary gland will undergo a further expansion during pregnancy and lactation. And once weaning is complete, it will sort of undergo a round of apoptosis to come back to a pre-pregnancy state. So this makes you immediately think that maybe there is a cell then that can undergo this significant expansion during pregnancy and then come back to a ground state. And it can do this several times. So it can do this for multiple rounds of pregnancy. So which brings me to a structure of the normal mammary gland, where there are stem cells and which give rise to the progenitor cells that can be either unipotent or bipotent, that will then give rise to the differentiated cells of the mammary gland, the luminal cells and the myoepithelial or the basal cells. So just like you have this in the normal mammary gland, what people know is that these stem cells, because they are long-lived, over the course of a lifetime will acquire mutations to give rise to what we know as the cancer stem cells that then form the primary tumor. And oftentimes when you have therapies that will just target all the cells of the tumor as one cell, so there is no difference in how the therapy treats each cell. You have the cancer stem cells which are refractory to this therapy that are left behind, that will then lead to relapsing of the tumor. And because these cancer stem cells are unique, what they can also do is they can survive in pretty harsh conditions. So they can go to a distant organ and form a metastatic cancer stem cell and give rise to metastasis in distant organs. Now, I'll just take a pause here to say that the cancer stem cell theory has been around for a really long time, actually. So I'm not going to go through all the papers here, although each of these studies and papers are really amazing to read. Go back and read. But what I'll point out is that some of the early studies, in fact, showed that tumor cells have stem-like properties. And one of the studies that I'd like to point out is by Pierce, where he showed that teratocarcinomas contain pluripotent stem cells, and he injected 1700 single cells to find that these these cells could give rise to differentiated cell types in the tumor. And when you took those differentiated cells and implanted them, they were not able to give rise to the tumor. This concept was then revisited back in the 1995 by John Dick's group, where he did this for AML, and subsequently by Mike's lab for breast cancer. So I also want to point out that although the clonal evolution model, which talks about the cell getting a mutation and that's the way a cancer cell forms, these two theories are not exclusive. They actually occur together in a cell. So especially for students, it's not that one happens and the other doesn't happen. They are probably happening simultaneously. So how can we go about finding these cancer stem cells? So what I do, and a large part of my PhD and my postdoctoral work has focused on getting tumor biopsies from patients, dissociating them into single cells, and then using fluorescent assisted cell sorting, in which we can coat the tumor cells with specific antibodies and then sort different populations and ask the question, which population will form a tumor in mice or in an in vitro system. So we can use either an in vitro organoid assay, or we can use immunocompromised mice. So I'll just show you one example where we did this. Oh, sorry, before that, yeah, I'm going to tell you also a little bit about the system we use and then go to the example. So we get these tumors from surgery. Oftentimes they are very small and we don't get them as often. So what we do is we implant the tumor in immunocompromised mice. And then you can see over here, the mouse has tumors and we can get these tumors back. And as I said, dissociate with single cells and then use them for whatever experiments we want to do. We can knock down a gene, we can CRISPR something out, we can do serial dilution assays or transplantation assays with them. One of the important things that we do also is we put them back in the mouse, so we have a renewed supply of these tumors, and every time we don't need to go back to the patient. And the reason for doing all of this is because in the mouse, the tumor cells maintain the heterogeneity that we see in patient samples, which oftentimes gets lost if you put them in a 2D culture system. So shown here is one of the examples of such a tumor. So this is a FACS plot, and each dot over here represents a single cell. This is stained for two antibodies, EpCAM, which is an epithelial marker, and CD49F, which is an integrin. And then the question we ask is, if we sort these into different populations or subsort them again based on different markers, which are the tumor cells? And we find that, in fact, the cells that are up here are the most epithelial cells are the ones that can form the tumor in mice. And as the cells lose their epithelial capacity, they become non-tumorigenic. So this is just one example of how we do things in the lab, but you can immediately see that there are several problems associated with this. First is that it is limited to the markers that we know. We can only test X number of cell surface markers in an experiment. And these markers tend not to be very specific. And when we try to sort these populations into subpopulations, it's difficult to, in fact, sort pure populations of cancer stem cells. So another thing that we've been doing to find molecular regulators is we try to do molecular profiling on these cells. And how we did that in the past was we sorted our populations and we did bulk RNA sequencing, and we get an average gene expression of all the cells. But in most cases, we cannot see what's happening at the level of a single cell. Fortunately, in the past few years, there has been so many available single cell technologies. So now we can do single cell RNA sequencing on these cells, and then we can actually see what is the heterogeneity within the cancer stem cell population. So that's exactly what I was doing. So in addition to sorting tumor cells by FACS, I also did single cell RNA sequencing on several patient samples and normal samples to really see what to see the tumor cells and the normal mammary stem cells at the level of a single cell. Okay, and then the question is now, how can we go about identifying the functional heterogeneity and identifying cancer stem cells from this single cell RNA sequencing data? So there are two approaches that I took. One is a more traditional approach where we know what the markers of our stem cells are, either from normal stem cell biology or from tumor stem cell biology. And the second approach is where we try to use computational programs to predict what is the lineage hierarchy within the tumor. So I'm going to give one example for each of these approaches. For the first approach is we knew that the normal mammary stem cells express a gene Thy1, and within that subpopulation in our human data, we saw that there was a subpopulation of Thy1 positive VEGF-A positive cells. And we wanted to, this population was very unique. So we wanted to see what is expressed within this population. And one of the top 5 genes was LMO2. And that was very interesting to us because LMO2 is a hematopoietic stem cell gene. In fact, what it does in hematopoiesis is it's required, and if you delete LMO2, the cells cannot undergo fetal hematopoiesis or adult hematopoiesis. And if LMO2 is expressed in the wrong place in T cells, what it makes them look like hematopoietic stem cells, and this ends up developing in a leukemia much later on in life. This gene was also one of the first genes used for gene therapy trial back in the 1990s. And what they found is that the patients who got this gene therapy trial ended up getting leukemia because LMO2 got activated in places that it was not supposed to be activated. So finding the expression of LMO2 in epithelial cells or breast cancer epithelial cells was really intriguing to us. So I went back to my xenograft system and asked the question, does in fact LMO2 do anything to the cells? And what we can do is we can take our xenograft cells and we can infect them with or transduce them with a lentivirus that can knock down expression of LMO2. We can put them back into the mouse and ask the question, what happens to the tumor? And what we found is that the tumors actually, they grow just fine. There's very little difference in the growth rate of the tumor. But when we look at the lungs, the mice that have the knockdown of LMO2 have significantly reduced lung metastasis as compared to cells that are expressing LMO2. And we can go back to the patient data and look at if patients who express high levels of LMO2, if they have an increased incidence of metastasis. And in fact, that was true. here you can see the red line is of patients that have higher expression of LMO2 in the epithelial cells as compared to patients in the blue line, which have lower expression of LMO2. And this looks at what is the probability of them developing metastasis in the future. What is the probability that they got metastasis? So this is post, um, this is the data we already know. So what we've gone on to doing is figuring out the exact molecular function of LMO2. I'm not going to cover that here, but I'm going to move on to our second approach, which is an agnostic approach. And this is, in fact, the thing that's most exciting to me, is because even without knowing what is the stem cell markers of which are the stem cells, we can predict which are the stem cells in any system. So for this, we collaborated with Aaron Newman's lab, he's a computational biologist at Stanford, and his graduate student, Gunsagar. And when I approached Gunsagar initially with all the single cell RNA sequencing data, we said, okay, let's look at what is it that predicts that a cell is a stem cell. And Gun went through every measure that was available and looked at systems that already where the hierarchy of a stem cell was defined, like for example the hematopoietic stem cell system, where we already know where the stem cells are, and looked at what is it that predicts that the cell is a stem cell. And one of the things that he found was gene counts. So just the number of genes that are, the number of different genes that are expressed at the level of a single cell was predicting whether that cell was an immature cell or a differentiated cell. And if you in fact look across differentiation, so you look at all the way from totipotent cells to differentiated cells across mounts ontogeny, what you find is as the cells are becoming more and more differentiated, they are expressing fewer and fewer different genes. So its transcriptional diversity of the cell is decreasing over differentiation. And this sort of makes sense because if you think of cells, as they are becoming decided as to what they want to be, they are closing off parts of their genome. And my next slide actually shows an experiment where this is a in vitro system looking at mesoderm differentiation. So this is human embryonic stem cells going all the way to sclerotome, and these are ATAC-seq peaks and the total signal. And as you can see that the peaks are decreasing over time, which means that the chromatin is getting more and more closed. And if you look at the number of gene counts too, it's decreasing as the cells are becoming more differentiated. So Gun and Aaron together developed a program, CytoTRACE, which can then predict stem cell hierarchies from single cell RNA sequencing data. And I'm going to show you a few examples over here. So on the left is shown is what I mean by ground truth differentiation states is things that we know from decades of work from several labs with lineage tracing and transplantation. So this is, we know that these are the stem cells and these are the more differentiated cells. And on the right are predictions by CytoTRACE. So CytoTRACE can accurately predict stem cells without any prior knowledge of what the stem cell was. This is not just true in mammalian systems, but it's actually true across other systems such as zebrafish and C. elegans. So again, on the left is the ground truth differentiation states, which is basically from experimentation, from work from several labs put together. And on the right is the predictions from CytoTRACE. So this is very exciting because in tumor cells, often the hierarchy is, it's very hard to predict hierarchy and it's often messed up. So if you think of our cancer stem cells that come from stem cells and acquire mutations as the undifferentiated cells of the tumor, and think that we can now apply CytoTRACE to figure out which are the cancer stem cells and what are the genes that they are expressing. And we did that with our single cell RNA sequencing data. So this is the prediction from CytoTRACE. So CytoTRACE is predicting that these cells here, which are colored in red, are the less differentiated cells of the tumor. And the ones that are in blue are the more differentiated cells in the tumor. And what we had also done in the lab is we had done an independent shRNA screen to look at what affects tumor growth. And we found that the genes that are affecting tumor growth were in fact expressed highly in the immature cells that were predicted by CytoTRACE. And there were also many genes that had previously not described in the context of breast cancer, or in fact, any cancer. And one of those genes that was GULP1. So I focused on GULP1 to go on with the validation because of several reasons. One, it is highly conserved across species. It was involved in this process of endocytosis and phagocytosis, which in the cells is a process by which the cargo or cell survival signals are transported from the cell surface to the nucleus. And an important function of cancer stem cells is that they prevent trafficking of certain receptors and certain drugs into the cell. And last but not the least was that GULP1 is expressed in other systems in immature cell populations. So for example, going back, this is the hematopoietic stem cell system. As we saw in Camilla's talk, you have the HSCs, which give rise to the differentiated cell types in the blood. And you can see that the expression of GULP1 was highest in HSCs, very, again, similar to our LMO2, which is also involved in HSCs. So we decided to go back and knock down GULP1 in our system. And we find that, in fact, when we knock down GULP1, it significantly impairs tumor growth in contrast to LMO2, which was not doing anything to tumor growth. And this is just one example, but as I said, most of the genes over there are understudied, and we need to look at what they are doing in breast epithelial cells. But I'd like to just conclude by saying, bring this talk together by saying that the tumor cells as we know it are not all the same. They are heterogeneous. And within this population, we have cancer stem cells. And even within the cancer stem cell population, there is heterogeneity, where we see that there are LMO2 positive cells that are specifically involved in metastasis. And we have the GULP1 positive cells, which are regulating tumor growth. And in the future, what we want to do is figure out what exactly are these LMO2 positive cells? How are they deciding between being GULP1 positive and LMO2 positive? And what is the function of GULP1 in breast cancer cells and normal mammary epithelial cells? So with that, I'd like to stop and thank everyone who has been part of this work. So most of this work was done in this building, largely in Mike's lab, where I was a postdoc before I came here, and to get in a very strong collaboration with the Newman lab and his student Gunsagar. Of course, many thanks to all the patients who give us their tumors and to my funding. Now I'm here and setting up my lab in Biomed, third floors. Please feel free to reach out to me if you have questions, want to discuss more, or generally want to talk or take a walk. I'm here. Yeah. With that, thank you.